Hello Year 9, we are going to be looking at genetic engineering. So this is a triple lesson aimed at triple students, and it's moving on from the work we've done on evolution and Mendel being the first person to talk about us inheriting two characteristics, and also we've looked at the structure of DNA. We haven't looked at DNA synthesis yet, or protein synthesis yet. But we will do that. But this is a two-part lesson. So today we're just going to be talking about uses of genetic engineering, and a very specific one that you must know for the specification. And the next one we're going to look at is ethics surrounding genetic engineering. So as you can see, I've got a picture of some animals there, I've got some piggies, and we have also got a cell with DNA and a picture of a plant. So it's kind of all-encompassing, this genetic engineering. So we're first of all going to look at what the words mean, but then we're also going to look at prime examples of how we can use it. And then in the next lesson, we're going to look at whether it's ethical to do so. So let's get cracking. So I'm just going to start off with these two questions to think about. What is genetic engineering? Now, you have probably heard this before, or you've heard of genetic modification. And then if you don't know anything about it, think about what these words mean. So break them down. Genetic, what does that mean, or what is it to do with? And engineering, what does that mean, and what is it to do with? So have a little think about those. You might want to pause this. I will go through the answer, like the specific answers, but you probably have heard it somewhere before, or something to do with something. Anything, root around and find anything in your brain, because that is how really good learning happens, if you try and recognise it. Okay, what we need to get out of this lesson is some advantages and disadvantages of genetic engineering. Now, that is going to come in the next lesson, but you might be thinking of these. You might want to jot some of these things down as you go through. You need to describe the modern uses of genetic engineering. If you can explain what genetic engineering is and what it's used for, that's about a grade 5 or 6. For a grade 7, 8, you need to be able to explain how insulin is produced, and do your advantages and disadvantages, or compare different types of genetic engineering, and those will come up as we sort of round off the skills. So back to our first questions. What is genetic engineering? Now, this is just copied and pasted straight off the first search that comes up on Google. So genetic engineering is the process of using recombinant DNA, let's just say DNA, we don't have to worry about the word right now, technology to alter the genetic makeup of an organism. So that's the key thing that we need to get from this. Genetic engineering is about altering the genetic makeup of an organism. And remember, an organism is a living thing. So this is about altering the genes in an organism. So changing or manipulating the genes in an organism. And the title sort of explains that. So what do the words genetic and engineering mean? Well, genetic means to do with genes, or the genome, or the genetic makeup of an organism. And engineering means to invent, design, change, build. So that is essentially, if we break down these words, we know that engineering is changing or inventing or designing something to do with genetics. Okay, so I'm going to go through. The next few slides are fun. You don't need to really know any of these specifically. They're not on the specification, but it's just to show you what we use genetic engineering for. Now, remember, genetic engineering is altering the genetic makeup of an organism, okay? So here's the first one. This is a glowfish. So they were originally created to show the levels of pollution in rivers. They are just your regular zebrafish on the left. They have had a gene inserted from a jellyfish or coral that make them fluorescent. So they have literally had their genes altered to make them glow. Okay. They can reproduce. It's actually illegal to make them, to reproduce them yourself, but you can actually buy them, genetically or genetically engineered fish. I mean, think about how this fish might benefit us in general. Could be a nice nightlight. That would be good. Other fluorescent organisms, so we've done it to mice, cats, sheep, etc. So we are able to alter an animal's genes to make them glow, which is rather marvellous, but also horrifically scary if you think about it. Let's move on and have a look at another one. Super spuds. Now, obviously, when we're talking about organisms, we're also talking about plants, insects, you know, cats, dogs. We're talking about all organisms. And obviously, if we can genetically engineer or genetically modify food, we can, there's some real benefits there. So this is the potato. It has got 60% more protein per gram than a normal potato with a larger yield, which means they produce more potatoes. So the plant will produce more potatoes. A gene was used from a different type of grain, which makes protein storage. So they've taken a gene for protein storage, popped it in the potato plant, and therefore you have potatoes that have more protein, which is good, and they also produce way more, so it means that you can feed more people. And they've tested it on rats and rabbits, and they seem to be fine. Some issues with it is that people don't want to eat genetically modified food because we don't know the long-term effects. So when we think about advantages and disadvantages, and if we grow too many potatoes, we can feed them to livestock, which is kind of good. We get this sort of recycling going on. But there is worry that it might cause some kind of resistance to certain diseases and things like that. So, you know, all of these are going to have benefits and disadvantages to them. So just continually think about that. Golden rice. Now, this is the most common example that comes up. And I believe this one is on the specification. So they have engineered rice that they have removed a gene that creates something called vitamin A. Or the building blocks to vitamin A. So vitamin A can cause blindness. So vitamin A makes retinol. And retinol, which means that your eyes are able to distinguish colour, which is another reason why the carrots help you see in the dark thing isn't quite such a ridiculous myth, because carrots are full of vitamin A, and it's called beta carotene. The yellowy orange part of carrots are called beta carotene. Now, normally when I teach a class, I show them my excess beta carotene stores, because most of you know me, I drink a lot of orange squash. And once you've stored beta carotene in your body and you've made all the vitamin A you've got, but you've got tons of beta carotene, it deposits in the skin and makes you look slightly yellow. And I've got very yellow hands. Because it just deposits in your skin when your liver's full. Now, this was a good thing, because vitamin A can cause blindness and death if you don't have enough of it, a deficiency in it. And most of the children that suffer from vitamin A deficiencies live in countries where rice is the staple. So they produced beta carotene modified rice for these countries, but they actually found out it was quite expensive. And some people didn't want to eat genetically modified food, not that they were in a particular position to not, if they were hungry. So golden rice is a good example. We'll talk more about that. Spider goat. Not as good as it sounds. Well, no, it is as good as it sounds. It's pretty good. So they produce genetically modified goats to produce spider silk in their milk. That's a hard thing to say. So they got a gene from a spider that produces the silk, and they inserted it in the milk, the genes that produce milk in goats, so that they will produce an extra protein which can be extracted and spun into spider silk thread. Now, this is good because spider silk is so strong when it's wound together, stronger than steel, lightweight, very elastic. It has a range of temperatures it can hold between, so it's really, really good. And they were looking at using it to replace ligaments, make waterproof clothing, bulletproof clothing, etc., because it's so strong. Obviously, there are some ethical issues here, because the goats weren't allowed to leave where they had been created. So most of them were euthanized. So these lives were created to be destroyed. So we've got to think about whether that would be ethical or not. And I like this little thing down here. It says spiders cannot be farmed as they're cannibalistic. They eat each other. So they had to use another animal to make the spider silk. So because you can't farm spiders if they're eating their own stock. Let's move on. Aqua Advantage salmon. This is a fish that humans haven't eaten yet. And what they've done is they have taken a gene which controls a hormone from one breed of salmon, put it into the other. This means that you can grow larger salmon, therefore you get more meat. Therefore you have to produce or farm less salmon because you're feeding as many people. We don't know the effect it would have if they escaped, but you can see the size comparisons there. But we don't know whether that's safe for humans to eat, because obviously it's a hormone, and a hormone is a chemical messenger, so we don't know the full effect that that would have on the human body yet. I mean, could it cause aqua advantage humans, which is worrying. This one I love, the venomous cabbage. Okay, so a cabbage that has been genetically engineered to include the venom from scorpions. Anyone want to eat that? I mean, not many people want to eat cabbage as it is. I quite like cabbage. So this has got venom in it, and so the point is it stops things like caterpillars and slugs eating the cabbage while it's growing. So it's not apparently damaging to human cells, but we can't guarantee that that toxin won't necessarily mutate, or it would go into biodiversity wise. If we're thinking about food chains, if I ate that cabbage, and then my waste, or the toxin that was in my waste, ended up in the sea, could that then affect the biodiversity of the sea or the food chain, like the build-up of toxins? Bioaccumulation is the word I was looking for. Potential. Flavour saver tomatoes. Okay, so flavour saver tomatoes. Tomatoes are probably the most genetically modified food, apart from certain flower crop, not flower as in flowers, as in, although they are heavily genetically modified, flour like wheat and grains. Probably the most common, and they put them in tomatoes to extend the shelf life, and it basically slows down the rotting, so they're red for longer. The other way we do tend to get tomatoes to ripen is that we spray them with ethylene, which affects a hormone which causes them to ripen at the right time. So we pick them when they were underdone, when they're green, or you can pick them when they're red, and then you can genetically, if you've got a genetically engineered plant, it slows down the decay. There we go. It was the first GM food to be sold in the world. Very interesting. And the banana vaccine. They have vaccinated bananas. No one's vaccinated a banana, Mrs. Corbett. So they have genetically engineered certain fruits and vegetables to deliver vaccine. The banana was the most successful. So they insert the gene into the banana. Its cells start to produce virus protein. So a part of a virus that's not going to infect you or harm you, but it enables you to produce an immune response. So this is kind of good that we're talking about this now when you're talking about pathogens in trilogy science. So only a small part of deactivated or dead part of a virus needs to be in your body to trigger an immune response, and then you will become immune because you've already created the antibodies, etc. So in theory, you can deliver vaccines through eating a banana, which is preferable to having polio squirted onto your tongue, or on a sugar cube as they used to do it, or an injection to some people. So potentially a really, really good thing. We don't know the effect that might have, and we also know that some people are very averse to protecting themselves against these diseases, or anti-vaxxers. So it's an interesting one. These haven't been generally released, but very interesting. And bananas generally are all clones of one another. So when we talk about cloning, we're going to get into bananas, because most of the bananas you eat are clones of each other. So if you're cloning this vaccine, it's really easy to spread vaccinations. Here's some others that we might not have covered. So we genetically modify crops being resistant to disease, or being resistant to a certain herbicide. So you might be spraying an area of crops to kill the weeds, and you would modify your plant to not be affected by the spray, but the weeds would die down. Reduce competition. Do excuse me, everybody. And yeah, we've had glowy animals. And look, if you look at the size of corn, the size of corn on the cobs has increased dramatically in the past 5 years due to genetic modification. And this is the one we're going to focus on, is insulin. You need to be able to draw and explain, or do a flow diagram of the process of manufacturing insulin. Let's do it. Now, the reason why I've got a picture of piggies is because the previous way to save a diabetic's life was to use insulin from the pancreas of a pig. Now, just to go into a little bit of detail, if you don't produce insulin, which comes from your pancreas, it means you can't remove excess sugar from your blood. And that is a disease called diabetes. And type 1 diabetics need to inject themselves with insulin, because if their sugar levels go too high, they can get hypoglycemia and die. So really, 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 really important. I'm just quickly getting the dates from my book. So insulin was first used for treatment for diabetes in 1922, and it was Mr. Banting, and I'm pretty sure he managed to isolate insulin from pigs, and he went onto a ward where people were in comas because of this hypoglycemia and they were dying. And I'm pretty sure, the first boy to receive it was a 14-year-old boy who lay dying in Toronto, so in Canada. Unfortunately, the boy had a really severe allergic reaction, and so they cancelled the testing on it. But over the next 12 days, they worked to improve the extract. They used oxes initially. It was completely successful, no side effects, completely eliminating any sign of diabetes from that moment. And what they did is they went to the large wards where people were mostly comatose, and they just injected them, and one by one they started waking up, which was absolutely amazing. So since 1922 we've been using animals, oxes, pigs, etc., farming animals, for this purpose of using their insulin. The problems with it is it's not human insulin, so it doesn't necessarily work as effectively. You can't necessarily test the efficacy of each batch. And obviously there are ethical issues with storing animals for medicines. So we're going to look at how we can genetically modify cells to produce insulin. And this is how most of our insulin today is manufactured. So what we do is we take the DNA from a human cell. Now, this has to be a healthy human cell, and by healthy I mean a cell that can code to produce insulin. So, for example, I do not have diabetes, so I have in my cells the gene for producing insulin. My sister, who is a type 1 diabetic, does not have a gene that produces insulin, or she does have it, but it is faulty or mutated and doesn't work. So we would find the piece of DNA, we would find the gene for producing insulin, we would use an enzyme. So this says lysozyme enzyme to cut a restriction enzyme to cut out the gene. So you can see that little red part here. So we would cut out that part. I don't know why the cutting out bit, the enzyme's a bit further away. And then what we would do is we would get a bacterial cell. So we've got our human bit of DNA, we've cut out the gene, pop that to a side. Then we've got a bacterial cell. And remember, bacteria have plasmids, those rings of DNA. So we get a lysosome enzyme and we cut out that ring of DNA. Okay, so the restriction enzyme is going to cut the DNA out of the plasmid, cut a section out. We're going to insert the gene for making insulin into the gap where the bit of plasmid DNA has come out. And then we're going to use ligase enzyme to join the insulin to the plasmid, the insulin gene to the plasmid. Then we are going to put the whole plasmid with the insulin gene back into the bacteria. Then we're going to put the bacteria into a little pot, lovely warm 36, 37 degrees, and we're going to allow it to multiply by a binary fission. So that means 1 will divide into 2, divide into 2, 2, 2, 2, 2, 2, and it's mitosis. And every single one of those bacteria has a gene to produce insulin, so they're going to be producing insulin. That insulin can be removed, it can be purified, and then humans can use it. And we call that gene splicing because we've taken a bit of DNA out and put it back in. So that's called gene splicing. We would also, this is genetic engineering, it's genetically engineering insulin, but we would say that the bacteria has been genetically modified. So if you get confused with the terminology there, modified means it's happened, engineering means that it has been changed. Okay, 3 questions. Now, this is very specific detail on a couple of these. I'm going to end here and then we'll do advantages and disadvantages on Tuesday next week. So you've got to describe the role of the following enzymes. So we've got restriction, lysozyme, and ligase. Okay, so the restriction cuts out. That's what it does. It cuts out the gene that you need. The lysosome removes the plasmid from the bacteria, and the ligase pops the bit of DNA into the plasmid. 3 reasons why bacterial organisms are good to use in this process. Well, they don't have complex nervous systems, so there's a lot less ethical, because they're single-celled organisms, there's no ethical dilemmas here. They're very simple organisms. They reproduce incredibly fast, like incredibly fast, so you can produce a lot. And they are actually producing human insulin, not animal insulin. So they're producing human insulin, so the side effects are very, very low. And then this is what takes us to the really higher end, where we compare and contrast. So when we compare, we look at similarities and differences between producing insulin from genetic engineering and extracting from animals. So I would, if I were you, to complete this question, is to do what's similar, what's different. Okay, so similarities is they're both producing insulin, which can be purified and used by humans to reduce the amount of glucose in the blood. The difference is that you are using a living single-celled organism or a multicellular organism in the terms of the animal. Differences are we're producing human insulin, animal insulin, so therefore we can have different side effects. So try and organise your work so that it's quite simple. I'm going to put some work on Doddle. Have a go at it. See you later, guys. Bye.